Okay, well, good afternoon, ladies and gentlemen. It is my great pleasure to welcome you to the Edinburgh lecture of the Society for Reproduction and Fertility 2006 Sex in Three Cities lecture series. Those of you who've been to these lectures before will remember that they were established to promote not only the Society, but also reproductive biology research here in the UK. This year we are really fortunate and really privileged to have Professor Graeme Martin, who agreed to be our Sex in Three Cities lecturer. Professor Martin is the Winthrop Professor in the Department of Animal Science at the University of Western Australia in Perth, Australia. Graeme grew up on a sheep cereal farm in Western Australia. He did his degree in agriculture and his PhD in reproductive biology at the University of Western Australia, and then worked for 2 years at the INRA research station in New Zealand, and for 3 years at the Medical Research Council's reproductive biology unit here in Edinburgh. In 1986, he returned to Perth to take up a joint position as a lecturer and a researcher at one of the CSR IRO facilities. He became a full-time member of the university staff in 1996, and by 2001 was promoted to professor. His research has focused on, firstly, processes through which environmental factors affect reproduction, and also the reproductive biology and technology in diverse species, including emus and ostriches. So we are really looking forward to hearing Professor Martin's lecture this afternoon, entitled, intriguingly, The 3 Ps of Reproduction: Pheromones, Food, and Photons. Not necessarily in that order. Professor Martin has indicated that he is willing to answer questions after his talk, and Apiwat and Billy, who are sitting on the front row here, both have microphones. So please could you wait for one of them to get to you before you ask your question, because the lecture and the question and answer sessions are being recorded. Those of you who've been before will know that a feature of these lectures is that we have a wine and canapé reception immediately afterwards. So I'd like to invite you all to join Graeme and enjoy some wine or soft drinks and canapés immediately afterwards. So without further ado, I'd like to invite Graeme to deliver the 2006 SRF Sex in Three Cities lecture. Thank you.
Okay, this is working. First stage passed. Thanks, Cheryl. A great introduction. It's, I'm absolutely chuffed to be invited to give this. It's one of my favourite science societies in the world, and I have great memories of SRF meetings over many years. And I have many friends around the UK, and it's great to see some of them here today. And I'm going to try and tell you a bit of a story. And really it's about the story of my career. So it's about my journey in science. And the way I'm going to start is to go right back when I was a PhD student, when I had this sort of basic question that it just simply personally intrigued me. How does the reproductive system respond to changes in the environment? At that time, there were people in particular in this part of the world that were focusing on photoperiod. And so that was one of the parts of the environment. But I was actually thinking more about animals in their environment, because they don't just see photoperiod, they actually see lots of different things. And they have to try and mix all this together to come up with some sort of strategy. So that was where I began. During my career, the other 30 or 40 years, there's been really 3 revolutions in our field of reproduction. And I'm going to talk about those today. The first is we discovered that a brain can make cells. When I was a student, that was not the dogma. The dogma was you were born with a certain number of cells and then you just lost them through your life. And you accelerated that loss if you drank too much. And the second was junk DNA. It's a concept that came and surprised lots of us and then went away. And finally, livestock management is that my own personal vision for livestock management has been completely reversed from where I began. and I'll explain that at the end as well. First though, we need to talk about animal models. So most of my work in my life has been done with this animal, the sheep. And that was partly, I suppose, because we have lots of sheep in Australia. My father was a sheep farmer. I'd grown up with these animals. I knew all about them, so that was a comfortable thing. Also, they don't bite. So I thought these were important characteristics. Okay, so it's an industry animal. So this is important because you can get funding from industry to do the research that is needed to advance industry and to investigate the animal's reproductive biology. And it's also a model. And this is a warning to all of you. If you Google model plus sheep, this is what comes up. Only in Japan would you have a model wearing a sheep costume and seeing it as a fashion statement. But really I'm talking about a biomedical model. And I need to justify this with a couple of things, right? The first is and the reason it's a biomedical model is because it's not that different to humans. So you might not like that idea, but it's actually very true. And I'm going to demonstrate a couple of things. The first thing I want to talk about is what I call the Montliggins legacy. So this is a Kiwi scientist, New Zealand scientist, Montliggins, and he was investigating the effect of glucocorticoids on maturation of the lung in the sheep fetus. And he made this leap to say, hang on a second, if it works in sheep, why can't it work in humans? And this now is standard practice in management of premature births around the world. So what we have is premature babies being saved. In 2015, 650,000 babies were probably saved because of this discovery. Why Liggins never got the Nobel Prize, I will never understand. A really amazing thing. Okay, the second thing, and it's more focused on my personal area of research, is about cycles. So let's look at the menstrual cycle. The menstrual cycle is controlled by the relationship between menstruation and this 28-day gap here. And there are a couple of hormones, as most of you know, that are really important. The first is estrogen, and it has a pattern during the cycle a bit like this, with a peak here in the middle. And this peak induces ovulation when usually one egg is produced. There's another hormone, progesterone, which looks a bit like this. And these two hormones in this relationship control what happens in the uterus and controls what happens in the brain and a few other things as well. So that's, in a nutshell, the simplest version of the menstrual cycle I can imagine. Right, let's compare it now to a really favourite laboratory animal. Okay, this is the estrogen progesterone pattern in a laboratory animal. And you have to admit it's a little bit different. Only lasts 4 days. Also, they produce 6 to 10 eggs at ovulation. So things are different here in terms of control of the ovary. While the woman is having one single 28-day cycle, rats are doing this, producing 60 eggs. So it's a very different sort of biology. Right, let's get rid of the rat. Now talk about this animal. This is estrogen in the sheep, 1 or 2 eggs. So a similar process of ovulation, similar estrogen profile, and a similar progesterone profile. So I don't know how much clearer I can make this, but the sheep and the woman are actually very similar, at least in this regard. Right, so we know lots about the cycle of the woman, we know lots about the cycles of rats, we know lots about the cycles of sheep and goats and cows, but there are other species as well, where we actually don't know so much. And here's an example. This is the white rhinoceros, and a student of mine, Annemieke van der Hot. So Annemieke loves rhinos for some reason, and she wants to try and save them. And of course, they're at massive risk because of poaching. The black rhinos probably disappeared. And so she wants to try and save them. The problem here is that the rhinos in zoos, where we might be able to have a captive breeding program and repopulate native areas once the problems have been solved there, don't reproduce very well. And then you say, right, let's solve that problem. But you don't know anything about the cycle. So how do you find out about the cycle? You can't study the zoo animals because their cycles don't work. So she had to go out to the field, and she spent 3 years in Africa following rhinos around, picking up their poo and analysing the hormones in the poo. So now she understands what the rhino cycle's like, what pregnancy is like, and so forth. So we're now moving towards a situation where we can start to solve the issues in zoo populations. So basic cycle research is still needed with lots of species. Right, back to my initial question, where I began. So it's all about the brain. And basically what happens here, it's about a basic reproductive physiology for you all, is the brain controls the secretion of these two hormones, LH and FSH. They control the gonads to produce sperm and eggs. And the gonads also produce the sex steroids. Now the sex steroids in the males, for example, are responsible for a whole range of these factors here, and one of which is odour, and that becomes quite important today because of one of my P's. In the female side, you have a whole range of quite parallel sort of functions, okay, but also odour. And so basically then, in terms of the sex phenotype and its behaviour and its chemical signals, it's really about these steroids in particular and this whole process. And this process is controlled by environmental factors, one of which is photons, one of which is pheromones, and one of which is food. Okay, you might think Australians can't spell food, right? Maybe you're right. Now, the critical molecule here is actually this one, the final common pathway, GnRH. So GnRH is a decapeptide, and in my days as a student, it was actually discovered and described for the first time by purification from 9,000 hypothalamic sheep, I think. And it resides in this part of the brain, down here in the primitive part, the hypothalamus and preoptic area. And basically then, all inputs travel via this molecule to get to the reproductive system. So everything about the physiology of the animal that the brain measures, everything about the external environment that the brain measures, comes in through here, and that goes down to control our reproductive system. Now, right from the very beginning, many scientists around the world realised that this is actually a critical point, a control point that could be manipulated as well for blocking reproduction. So if you took out GnRH, gonadal activity would subside and give you a form of contraception. And this is what's happening here with this animal. This is the black-footed rock wallaby. And it lives on rocky outcrops in Australia. Most of these rocky outcrops are in amongst farms where they get lots of extra food and lots of extra water. And we have an overpopulation problem. The overpopulation problem makes the farmers very angry, but also the animals then begin to starve, and we have serious ethical issues associated with them. So we need to manage it by population control. And Nicole Willers, another student of ours, has been looking at this and using deslorelin, a GnRH superagonist implant, to control populations here, and then studying the effects on their gene flows, their migration strategies and so on. Right, back to where we were. Let's look a bit more detail at this system. So we're talking about the hypothalamus and pituitary gland, the real base of the brain and the place where we have this communication with the body. And diagrammatically, it's a bit like this. This is the pituitary gland, this yellow blob here in my schema. And there is a series of blood vessels that starts in capillaries and ends in capillaries, a portal system that goes through here. And this is connected to the GnRH cells. These cells are simply amazing cells, right? Now, okay, it's my favourite cell, but it's really an amazing group of cells. These cells begin in embryonic life way out here in the nose of the animal. They then migrate in here into the base of the brain, then along the base of the brain to establish this connection. No other cells do this. And if you block that process, everything goes wrong. And this is what happens with Kalman syndrome, for example. The other amazing thing about these cells is that the cells don't act independently of each other. Somehow or other, they are synchronized. So they all fire together to release their hormone into the bloodstream. So blood comes in here, and if you take blood samples and measure it in here, such as some clever people like Ian Clark can do, you can see this phenomenon. You can see the pulses of GnRH as these synchronized cells fire. And these pulses are absolutely critical. The frequency of the pulses is the driver of reproduction. When the frequency is high, reproduction is ramped up. When the frequency is low, reproduction subsides. And so clearly this is an important thing to look at. Coming out of the pituitary gland, this signal brings out LH and FSH, which controls the gonad. So clearly then, if you want to understand this system, you need to understand these cells. The problem is, of course, that they're deep inside the brain of the animal. So you can kill the animal, and you can look at them. So and they look like this if you have a sort of special dye, and you can study them and you can see which ones are activated, which ones are not activated. But this is quite limited as an approach because basically the animal's dead. So you can't do many types of experiments that we need. You can do, like some people do, and measure GnRH here in the bloodstream, And that's also really complicated. The surgery is difficult. Only about 3 people in the world have ever really mastered it properly. And it's expensive and complicated, and there are ethical issues and so on. So it's really just not the best thing to be doing. But the joy of it is you don't have to do any of that, because you can measure LH in jugular blood, take a little tube in here in the neck, and you get a bioassay of the activity of these cells. So by measuring this frequency, you measure this frequency, and you know what these cells are doing. And that in a nutshell, ladies and gentlemen, is my career. Right, so let's look at pheromones. Pheromones are chemical signals, and they can inhibit and stimulate reproduction. So one of my students has been studying these animals. This is the African wild dog, and this is Leanne van der Weyde. So the wild dog is fabulous, right? It's an endangered animal, mostly in sort of southwest Africa, and she worked in Namibia. What happens in zoos here is that, in fact, they reproduce really well in zoos. We'd like to find a way to stop that from happening. But the fascinating biology is that the dominant female in the pack sends out a chemical signal that stops the other females from cycling so they don't get pregnant. Then those females look after the pups while she goes out and hunts. So this is an amazing bit of behaviour, controlled, we think, by a pheromone signal, though the evidence is still a bit shaky. So she also followed these animals around the bush, picked up the poo, and has been measuring the hormones. Right, what about pheromones that stimulate reproduction? This brings me back to my sheep. So let's look at the brain signal in males, and this is my brain signal, don't forget, the LH in the jugular bloodstream, in 2 different males here. I've given you a couple of examples, over a period of 12 hours. And when you look, you get a pattern like this. Male sheep don't have many pulses. It's usually about between 1 and 4 every 24 hours. So it's a low frequency. Here, I introduced a cute female sheep. All right? And this is what you see. Basically, before you can even take the first blood sample, the brain cells of the male have responded to the smell, and you've had an increase in this signal, which drives testicular function. Right, well, what about the females? So here's my brain signal again over a period of hours, right? But here it's a slightly different experiment. Here what I've done is I've placed familiar males, what we call familiar males. I've placed males with females for 3 months. So they become very familiar. And then what I did was, this is what we see when that happens, okay, a nice basal secretion rate. Then what I did was I took one of the familiar males out, walked out of the room, waited 15 minutes and brought back into the room, okay? And this is what happened. Nothing, okay? Really boring, this guy, obviously. And this one, I put in a novel male that she had never seen before. And this is what happened. Now, I can tell from the giggles around the room, right, that you're thinking about pubs on Saturday night and relationships between people and so on, and I'll let you speculate in that area. But quite clearly here, this issue of novelty is important. So what does that tell us about the biology that's happening in the background? What it tells us is we've got an issue here of recognition and memory. So we looked in memory centres. The memory centre of the brain is an area called the hippocampus, like this in the sheep brain, and there's a white band through here. I'll put dotted lines to show you what it is, called the dentate gyrus. So we looked in here to see what happens when we introduce the smell of a novel male. And what we find is that cells divide. So this is cells stained with BRDU, and BRDU is incorporated into the cell when it synthesises new DNA. So cells that divide obviously have to synthesise new DNA. So you get 2 cells where there used to be 1, and they're both coloured. So you can tell what's happening. So what we find now is that the male odour stimulates cell division in the memory centres. Okay? We think that the new cells make a new memory. In fact, we think that the cells migrate through to different parts of the brain and establish new pathways, which establishes what we call an olfactory memory. The absolutely wonderful part about this is this exact same process in exactly the same part of the brain. Cell division in the hippocampus happens when a female sheep gives birth to a lamb and learns the smell of her newborn lamb. So she's getting double bang for the buck out of this part of the brain and this process. Right, so that's pheromones. Let's now go to photons. So photons enter the brain through the eye. The information travels down the optic nerve and into the hypothalamus. At this point, it goes in 2 different directions. One direction is towards a 24-hour clock, and it keeps the 24-hour clock in sync with day-night patterns. The other direction is to the pineal gland. Pineal gland produces melatonin, but it only does this at night time. So therefore, the melatonin signal tells the brain of the sheep how long the night is. So we have here a measure of night length. And then the brain of the sheep looks at these two things, the 24-hour clock and the night length, brings them all together and can decide if it's autumn or spring, if the nights are getting shorter or the nights are getting longer. And this, of course, tells it the season. This signal then travels to the reproductive centres. The consequence is that we get in the autumn we get a high frequency of the signal, and in the spring we get a low frequency of the signal, and that's about breeding seasons. Right, just to show you it's not just a joke, here's some data, my brain signal over a 12-hour period in a female sheep. Lengthening nights, so autumn, winter, and this is the signal we get. A high frequency of GnRH pulses driving the reproductive system. And of course that means we have the breeding season. Under shortening nights, what we get is this. So this is a low frequency of our brain signal, which allows reproduction to subside, and so we have the non-breeding season. So it's a really simple, well, it's a complicated process happening inside the brain, but the output is really, really simple and very elegant. Now up here, Gerald Lincoln and David Hazelrig have been looking in this part of the brain here and looking at what goes on during these phenomena. And I'm going to show you a little cross-section of tissue through here. This is the third ventricle here in the median eminence, and you can see these little bright green dots, at least I hope you can. These little bright green dots are indicating cell division. And what we've discovered then is we've got new DNA, new cells, right? And they have argued that these cells are actually little niches of stem cells and are involved as an annual clock, part of a 12-month process going on in the brain to keep the reproductive process synchronised with annual changes in seasons. Really exceptional work. Right, this whole phenomenon doesn't only happen in sheep. Let's talk about this bird. So this is the emu. This is a male emu, and these are the chicks. And this is a typical photo, really, because in emus, the male incubates the eggs and looks after the chicks. And this is Eric Malecki, who did a lot of work with us on this, and he's actually checking underneath a male emu on the nest to look at how many eggs are there. So what happens here, though, is that once again, this is a photoperiodic bird. This bird responds to the length of the night. And it's really interesting because it actually is focused in its natural distribution around the tropic. So with the change between summer and winter, it's probably only about 2 hours. And yet it dedicatedly follows these 2-hour changes in season. Except for some occasions when there's a summer thunderstorm, and then it advances the egg-laying season through processes we have no idea how they happen. Right, so let's summarise where we've been, pheromones and photons. This is my first revolution. Brain cells can divide. I mean, this was unbelievable when I first read it, because it was just against all dogma I knew. Brain cell division is linked to the control of reproduction, so it's not just something that happens, it's actually a controlling process. Implications. We have new thinking about how the brain works, that's pretty obvious. We have this idea that the brain now has stem cells, and we now are confronted with the possibility of brain repair with diseases like dementia. So this is a really exciting area of research. Right, food. Okay, let's get rid of the pH. So food, the third of my F's. Food is the brain signal. Let's look at the brain signal. Here it is, blood LH in 96 hours in a male sheep. This male sheep is on a constant diet, so it sits there at constant body mass, and it's about average body mass and body weight and body condition. And it has 4 pulses in 96 hours. These blood samples are taken every 20 minutes for 96 hours, which is why we have PhD students. Right, same situation here in this period, but here what I do is I provide a food supplement, and this is what we see. So basically, I provide a food supplement, and in fact, statistically significantly, by 6 hours, the brain has decided that the food supply is good, and therefore it should gear up reproduction and start to drive, in this case, the testis. So this is the process we're talking about, brain controlling testis, in this case looking at food responses. I just need to change this diagram. This is fairly classic. We always put the brain in a superior position, don't we? Well, I think we should just change that and put the testis where it belongs. And so, with the testis in this superior position, and that's because I want to look inside the testis now rather than inside the brain to see what's happening. So, mature male sheep on these different diets, high and low, for 9 weeks, and we see an increase in testis mass. Now, I don't know, men, if you've ever weighed your testes, but as you can see, they're bigger than ours, right? So, this is important because every gram of testis produces a certain number of sperm per day. And so therefore you would expect sperm production per day to be increased, and that's what you see. So billions per day, this is the numbers. Okay, that's pretty straightforward, but there's an interesting phenomenon here we can find out by looking at these two sets of data. This is an 86% increase in testis mass. This is a 250% increase in sperm produced per day. So it's not just an increase in mass driving this, there's something else happening. It's what we call spermatogenic efficiency. So each gram of testis has actually become also more efficient in terms of the number of sperm produced. Question, how does that work? Right, this brings me to this phenomenon. This is what we've all grown up with in textbooks, okay? DNA is transcribed into messenger RNA and then translated into protein. And I've called this big RNA because you'll see why in a second. So this is what we know. Now we also know that little bits of RNA called small RNA are also produced. And these sort of float around the place, come in here, bind to the big RNA and block it. And this is now microRNA, pRNA, and a whole range of these small molecules, in fact, thousands of them. So, okay, the next slide is complicated. Right, it's complicated. Are we ready? Basically, I'm trying to link then nutrition to reproduction with what's happening in the testis. Okay? And here it is. So, nutrition in, sperm out, and wherever you see a little yellow arrow here is where these microRNAs, these small RNAs, are involved in the response to nutrition. And they control the flow of spermatogenesis, and they control the rate at which sperm cells die before they're produced. And that controls the efficiency of sperm production. And this fantastic project is run by Yongjuan Guan. She was a student of mine, just finished last year, and she's now working in Philadelphia. Right. So that shows you how food can stimulate the reproductive system, but unfortunately food doesn't always work that way, because if you have too much you have an issue. So this is Gabi Flacker, and she works on the pygmy hippopotamus, this little guy. This is an adult female pygmy hippopotamus. So they're not very big. They're endangered. They come from West Africa, like the Ivory Coast. And what you see here is a little hippo with a very fat neck. Okay? How do I know it's fat? Because Gabi did some camera trapping in the Ivory Coast just before Ebola. And this is one that she trapped on her camera trap, and you can see how skinny the neck is. It turns out that these little guys reproduce badly in zoos because they're overfed. And so zoo management's just been a little bit too generous. And we now have to find out new ways to manage their diet so they start to cycle properly, and then we can use them perhaps as a resource to repopulate the endangered species in West Africa. So that brings me to revolution number 2. Some DNA is genes, but lots of DNA is used to control gene expression. And this is really something I never imagined when I was a student. And it's a complete revolution. New thinking about how DNA controls cells, and new options for fixing malfunctioning tissues. So once we begin to understand these processes, we may be able to find ways to intervene to fix things that have gone wrong. Okay, let's go back to where we began as well, with the industry animal and biomedical model, because what I want to do now is go back to the industry side and look at what I've talked about and how we're applying that to an industry situation. The background of this is this. Animal products around the world are being viewed differently by consumers. Basically, our society's attitudes to animal products are changing. And I summarise this in 3 words: clean and green and ethical. So clean clearly means less dependence on hormones and drugs. This is what consumers are demanding. Green means ecosystem care. Ethical means thinking about animal welfare. None of those things will surprise you. But they are market forces. How do we know that? Well, in terms of product quality, we used to traditionally think about meat in terms of sensory perception, taste, tenderness and so forth. Maybe food safety. We would think about milk in terms of protein content and whether it's contaminated or not. Now, the origin of the food is a part of quality assessment. People are asking when they go to buy, where did it come from? How did it get here? This is important. It's particularly important for us because if we don't adopt these concepts, we risk our whole market. We are a food exporting country in Australia. We produce food for 60 million people and there's only 20 million of us. So we depend on export markets. And our export markets are people like you and the US and Japan, and they're very picky people. So we see this as the future of all of our own animal industries, but we also have a worldwide perspective that says, in actual fact, that should be happening everywhere, not just in our country, not just in yours. Let me show you how we can implement this. This is the breeding process for a sheep. Mating, a pre-mating period, a mating here, pregnancy, birth, lactation, weaning, and if you're a farmer, this is where you make your money. So, the first of these is nutrition. Where does nutrition affect the reproductive process? Damn it, it affects it everywhere. So this is a headache for farmers, right? Because food is not perfect year round. So how do you manage this process? So they have to make decisions about when animals are born in relation to food supply. Also though, we as scientists can say, okay, what can we do with these arrows here? How can we make use of these arrows to control the process, to improve it, to manipulate it? And so we're thinking very hard about that. So for example, we can control the proportion of twin sheep born simply by a 3-day supplement. 3 days costs nothing. And 3 days is enough to change a female sheep's brain from saying 1 egg to 2 eggs. Amazing. Photons. I have a colleague in Mexico who works with small goat producers in the semi-arid areas. They're very poor people and they'd be happy to have 25 goats. And they are always troubled by the issues of the timing of kid production. And now they can do it. They put the animals into a rickety old shed and they put a light globe in there. They change the length of the night and therefore can control the timing of reproduction, which helps them a lot with their profitability. Pheromones. What I've shown you here is an amazing phenomenon that can control the whole timing of these processes. So when you take female sheep and you introduce a novel male, all the females respond at the same time and they're therefore synchronized. That allows the farmer fantastic precision in the control of this whole process. Control here so he can reduce neonatal losses, control here for growth and milk, control here if we ever get to a situation where we can take advantage of fetal programming as a way to control productivity. Everything you see here is clean and green and ethical. So it fits our vision for the future. Right, this brings me to revolution number 3. We can control reproduction in animals by manipulating their environment. We don't have to inject them with hormones. Implications. Clean, green and ethical management of livestock is actually not just possible, it's actually available right now. It costs nothing almost. Anybody can do it. It's not complicated. And we are now pursuing the idea of application worldwide. How do we do that? Well, this is a question. I'm a part of an international network and we ask this question, right? In terms of livestock industries, can they help feed the world without destroying the planet? This is a question that should be on everybody's lips. We had a big workshop, we published a paper, and these are the major issues we described. Environmental footprint. So clearly methane pollution and other sorts of pollution need to be controlled. Animal nutrition, health and welfare. Clearly clean and green and ethical, but in other words. And these are factors that are about the image of the industry, but also about productivity. Every one of those 3 factors affects productivity. Human food fed to livestock. We currently feed about 1/3 of the total crops produced around the world to chickens and pigs and dairy cows. Should we do that? Genotypes not suited to environment. I don't know why we insist on sending Holstein dairy cows from a tropical environment to southern Africa where they die in the tropics. The relevance of livestock to local culture. So the do-gooder people who say, oh, we can solve the planet's problems by getting rid of animal industries, just don't understand how culturally deep some of these relationships are. Human nutrition, healthy eating. About 1 billion people in the world are starving and about another 1 billion are overweight. And there's an imbalance there in the way we distribute food and consume food. And finally, all of these things are what happens in industries and on farms. And you can't solve these problems by looking at one factor at a time. You can't sit in your white coat in some ivory tower and say, I'm going to work on this, and that'll be the solution to the problem. You've actually got to understand the interactions amongst these factors. And that means you have to understand farmers. Farmers have complicated lives. And until you have a farm and try to manage a farm, you'll never understand how complicated it is. So we need model farms. And so we have a network of model farms. One in Uruguay, for example, we've got partners up here, not farms, Devon here, Kerala, India. Malawi is not farms, but a cooperative. We have got Zhejiang province here is thinking about joining us. In fact, I think Northwest will be faster. This is us, the biggest one, obviously. Queensland, New Zealand dairy farm. So this is our farm here, and this is encapsulated in a single project in our university called UWA Future Farm 2050. Please note the Facebook page. I'd like you to like us. Right, so Cheryl, I'll come to an end now. Just to summarise then, this is where I began 30 or 40 years ago. And through this really amazing period of science and scientific development, I've seen 3 major revolutions. We now know that brain cells can divide. I mean, that's an amazing thing. We now know that some DNA is genes, but lots of DNA actually does other stuff to control cells that we never imagined. Watson and Crick certainly never imagined it. And then revolution 3 is we've reversed our thinking about livestock. When I began, we used to think that all livestock control was just done by injections. We're just making better and better injections. Now we're thinking, nah, we don't do that. Let's just reverse the thinking and use the environment to control the animals. Thank you very much.
Well, thank you, Graeme, for a truly excellent and inspiring lecture that I think exceeds all the expectations of the SRF Sex in 3 Cities series. So thank you very much. I'm sure there'll be lots and lots of questions because this covered such a lot of topics and so many broad areas. Who would like to ask the first question? Right at the back.
Hello, thanks for your talk.
Could you wait for the microphone, please, because it's being recorded?
Hi, thanks for your talk. I was wondering, I'm impressed by your experiments on olfactory memory. So I was wondering about a time scale, if you've got any data on time course, and particularly if the same process is affecting during aging, if you've done anything in this situation of aging, whether there is a reduction actually of the olfactory memory.
Okay, I didn't have time today to go into the details of this, but there are obviously time courses. In fact, the production of new cells happens within 2 hours of the smell of the novel male arriving, and the same happens for the newborn lamb smell with the mother. It's probably faster, but we don't know because it's technically difficult to get into there earlier because of the relationship with injections and the sacrifice of animals and so on. But it's at 2 hours at the maximum. It's happening before it. Then obviously somewhere down the line they have to forget. Okay? Otherwise a female sheep would have like 30 lambs following her around for the rest of her life. So clearly there's a process there of olfactory memory being created and then disappearing, probably through apoptosis. We don't know anything about that part of it. We know roughly that they remember the smell for about 3 months. It could be shorter, it could be longer, but that's a figure that we're all talking about as a rough estimate of the process. Does that answer your question?
Yes. So are you planning to do anything in aging at all?
In aging? No. No. Actually, it's a good idea considering my age. Yeah, good idea.
Do you know if the olfactory attractiveness, if that's the right term, is in any way associated with the relatedness of the ewes and the rams? Because there are data suggesting that similarities in MHC and such like can affect the degree of attraction.
Yeah. We've toyed with this idea quite a lot. We've got in Perth an expert on sheep MHC who's always been encouraging us to go in this area, but we just simply haven't got around to looking at it. We'd be sort of surprised if it didn't happen that way, and then maybe surprised if it did. So we're sort of tossing between the ideas. Part of the problem here is that the chemical signal is really complex. We have an image of a chemical signal being a simple molecule like in insects, and it's not like that. It's almost certainly a complex mix of fatty acids, because otherwise individuals wouldn't have different signals and therefore different memories. And so, and certainly in lambs, the smell is related to the genes of the lamb, not the genes of the mother. But that's about as far as we've got. Okay. But it's a really important question, yeah.
Thank you. There's a question just here.
Sorry, just out of curiosity, if the if the olfactory memory works, can you actually put like a female sheep into a barn that's have some normal male that's been living in there? Would that still occur?
Yep.
Like without seeing the male?
I don't know if you heard that. So the question is could you simply put female sheep into a barn where there's been males, novel males? The answer is yes. That would work. Depends on how long the males have been there to a degree, because the odour comes from the skin. And so what we often do, we want to narrow down the issue to smell rather than vision or sound, is we simply cut wool off the male and we use the wool. You rub it on the nose. Okay, so that's why we know that would work. If the males have been there long enough to leave wool and pheromones around. There's not many people working in this field, right? So I have some colleagues in Japan who work in this field that they use goats. And interestingly enough, goats can stimulate sheep and sheep can stimulate goats, which is really unexpected. But there's also risks of oxidation of the pheromone. So if you take a sample and it oxidizes, then we think that might make it better, but we think it might make it worse, and we just don't know. So there's just too many questions in here, plenty of room for scientists to get working.
I think there's another question just next Noel.
I think...
Oh, sorry. Would you mind waiting? Aaron's got one. Sorry.
Go on, Aaron, you go.
I don't know if this is working.
No.
So I just wondered if you had any ideas as to how we can apply things like photoperiod and pheromones into management and livestock production of cattle who are not seasonal breeders, and we tend to use things like AI quite a lot so they don't see the bulls.
Yeah.
What can we do?
Yeah, that's right. That's a vexed question, right? Is what do you do about species that don't respond to light? And cattle, many genotypes of cattle do respond to pheromones. I have colleagues working in Mexico again who use multi-purpose breeds, and they clearly respond to the male in terms of shortening postpartum estrus. So my view is that inside every animal is basically the same set of genes, right? And they're just switched on or switched off. And we've just got to find out what's going on. And then we'll have ideas about what we can do. So it's about looking at the genes. The difference between a Suffolk and a Merino ram is quite remarkable. You can have a Merino ram, you can have a Suffolk ram rather, in the breeding season, losing body mass. Okay, losing a kilo a week, but its testes will grow because it's controlled by photoperiod. And then Merino says, "You want me to lose? You've got to be joking, So my testes will grow when I'm eating well." So, and yet they must be how many genes can be different? Not many. Okay. So somewhere in that relationship between food and photons, right, there will be genes controlling a switch that says do this or do that, or do a mix. So I think that's where the answer will lie. But maybe also because cattle basically have a 1-year cycle, maybe it's not necessary anyway.
So I think there was a question here.
I want to ask one basic question is about how much difference between the sheep olfactory bulbs with the human one? Because you try to compare between the models.
Yeah. Okay. Good. I don't know.
Could everybody hear that question?
So the question is, do sheep olfactory bulbs and human olfactory bulbs, are they similar? Or they're basically similar in structure, okay, in general structure. They've got an accessory system and a main system in much the same way. The sheep olfactory system that's involved in pheromone responses is the main olfactory system, not the vomeronasal accessory system. And that's something that comes out of rodent literature. So in rodents it's the accessory system, in sheep it's the main system. And it goes in through the amygdala and so forth, and then down towards the hypothalamus through a whole heap of unknown connections. Does it happen in humans? Everybody's intrigued by the answer, aren't they? You just know, people saying, "Oh, does it work?" There was a really famous study done in, I think, Sweden about 10 or 15 years ago in a Swedish university where they had roughly 100 students, and they got the male students to wear the same T-shirt for 2 weeks without washing. And then they got the female students to select amongst those students for the T-shirt, well, to select amongst those T-shirts for the T-shirt they preferred. Very brave women, Swedish women. And then they did an analysis, and they found better genetic compatibility amongst the T-shirts that they preferred and their own genes. So theoretically, they'd have stronger, in MHC, stronger offspring in terms of immunology. And you think, wow, that's pretty amazing, isn't it? So I think there are reasons to think that in humans, basically the same biological processes are hidden away underneath, probably messed up by underarm deodorant and showers and baths.
Alan, I think you had a question.
Fantastic.
Yep,
It's actually on,
yep.
It's a superb talk, Graeme.
Oh,
It
thanks,
puts
Alan.
things into context that I think people don't think about. This constant problem in medical research, working with clinicians who want to treat a patient immediately and don't understand that a mouse is not actually a good model for a lot of the things they're working on.
Yeah.
But one of the things that Gina was just talking about is the fact that there was always the Whitten effect in rats and Bruce effect in mice. The Bruce effect, Hilda M. Bruce published it in the middle of last century. So it's still true, guys. Yeah? That if a mouse, female mates with one male, then you change the male, put it in the next day, she'll have bought that and then mate with a second male. Maybe this happens in pubs as well, I'm not sure. And of course the synchronization of menstrual cycles is known in sort of nurses' homes and stuff like that. It's presumably phenomenon. But is there a similar sort of phenomenon of female selectivity of male mates in other species?
There's a lot of that phenomenon in insects, some in birds and some in frogs. Not much evidence apart from the rodent literature in mammals that I know of, right? So for example, sheep, goats, cattle, you can't see a novel male inducing abortion or resorption or whatever. It could be because we haven't looked cleverly enough. But
I wonder if you have 4 rams together with a flock,
mm,
and one's a king.
4 rams, one's very dominant, causes abortion for the fetus of the others. I haven't seen any evidence for it, but it doesn't mean it doesn't exist, right? Maybe I've had my eyes shut. Yeah.
I think there's a question right at the back, just to keep Billy good and fit.
This is another question sort of related to humans, and it's probably a really basic question, but how actually is the human reproductive cycle influenced by photoperiod, or is it at all, or what do we know about it?
Yeah. It's funny how you keep asking human reproduction questions of a sheep scientist, right? So, but I'm crazy enough to speculate. So there's no obvious, really direct seasonal pathway like we see in birds and sheep and so on. So that really strict relationship doesn't exist. But there are lots more subtle effects. You've all heard of seasonal affective disorder. Seasonal affective disorder is basically us responding to our melatonin profile. So, we get cranky when the days are dark and dim and go forever. And so then we head down to Mallorca for a holiday. So those sort of phenomena are well tracked and traced and highly demonstrable. But I can't think of anything in the human reproductive cycle that no, I can't. He can't either. He's a guru.
I was going to ask you about the opportunities for feeding animals in order to achieve the attributes and the traits that we want in the context of homeostasis. So, to what extent do you think animals respond to a change in nutrient intake, which would affect metabolites and such like? To what extent do you think that we can continue with altered nutritional regimes and still have the desired benefit? Because presumably some of the benefit is associated with the animal adapting to a change in diet.
Yeah, it's a complex question, Cheryl. Okay, so we need to remember a little bit of the evolutionary history of our farm animals here. You know, before humans came along and took them out of the bush, they'd lived there for millions of years. And they were adapted to a cyclical, seasonal environment with periods of abundance and periods of starvation. And so their whole metabolic processes are geared up to handling that. And they're just, they didn't evolve to have the perfect diet 24 hours a day, 365 days a year. So on the other hand, so that's a normal, the cycles are normal on most big farms where you don't have a lot of intervention. Our experimental models, usually because we have to keep our end down because we've got blood samples and analysis to do, usually rely on acute models. So the work we do on nutritional pathways in the brain of the male sheep, for example, relies on me wandering in the morning and just changing the diet. And I give them 500 grams of lupin grain. And lupin grain happens to be just the perfect nutritional stimulus because it has no soluble starch in the grain, so they don't get acidosis or bloat, which would kill them. But they can eat this large sort of like chocolate bar, and then we can watch the responses. Now, it's pretty hard to imagine that sheep evolved a system where they would just stumble in the morning over 500 grams of lupins as they wandered through the field, right? So that's clearly like, it's almost like nutritional pharmacology. It's a bit unrealistic, but it allows us to figure out pathways. And that's a long-winded way of saying I really can't answer your question very cleverly. Okay? But I'm sure that they've evolved all sorts of pathways to respond to changes, in particular to energy balance, that we just haven't quite figured out yet. And because the rumen is such a big buffer and can convert protein to energy, it's quite tricky to figure it out. But they're certainly very responsive to energy supply. Everything we do about nutrition reproduction relationships in growth is about energy. And they're very responsive for milk production, for reproduction, for ovulation rate, for everything.
Have you looked at differences?
It's not working.
Have you looked at differences in response to attractive versus unattractive animals?
Did everybody hear that?
Have we compared attractive and unattractive animals? Is this a human question or a sheep question? So the answer is yes. Right? So we haven't looked at them in terms of the pheromone responses, but it's really clear that female sheep, for example, find some male sheep attractive and others unattractive. You can simply do preference testing and they'll demonstrate very clearly they think this bloke's ugly and this guy's cute. And what's more is the cuteness depends on how much wool they've got, which may be pheromonal. Okay, I don't know. But it's a really good question. But they certainly can discriminate that way. And sheep get bad press for intelligence, right? Anybody who's worked with sheep will say they're stupid. But at the same time, that's only because they don't do what we want them to do. And we have the same way of classifying dogs, don't we, right? And sometimes people. But what happens, sheep can remember 50 sheep faces, at least 50 sheep faces for at least 2 years. So they've got remarkable memory. Outside the olfactory memory, they've got this visual memory as well. And I think I struggle with 50 human faces. And so it's that process of assessment it requires. And I said at least 50 for at least 2 years because the guy who did the work, Keith Kendrick, got sick of testing, right? It could be a lot more.
Chris, maybe one last question? Graeme off the hook.
I need my canapes and wine.
Very nice talk.
Thank you.
So this is a sort of question that sort of comes off the back of that question as well, so there's experiments done with Japanese quail, whereas the case of you show a female, just lift the sort of divider, show the female the male, and she will go into, well, so start to produce an egg. Do you feel that there's any, so we talk about pheromones a lot, but do you think just the presence of a male visually, minus any scent, will also have the same effect?
That's a really good biologist question, that one. So how do I know it's pheromones and not images, not sound? Well, because we've tested. Okay, so basically, then the data is very clear. We can shave wool. Wool does the effect. We've shown, you know, pornographic movies to sheep, not of humans. Careful now. You know, sheep mating of rams, of boys, of girls, and so forth. They show some response to a movie. Okay, we can get just a little LH response sometimes to the movie, but basically, it's very hard for us to demonstrate anything visual or auditory that causes the LH response. If it does, it's very minor. It's really, I'm sure it's 90% smell. On the other hand, I'm not sure. I can see what a sheep sees and I can hear what a sheep hears. So, you know, it just could be that we've just not done the right experiment.
That's great. Well, thank you very much. I'm sure you'll all want to join with me in thanking Graeme again for a truly excellent Sex in Three Cities lecture. Thank you.